Welcome to SCigest, your fortnightly science from the Bioeconomy Science Institute.
Kia ora koutou, and welcome to another episode of SCigest. I'm your host Tom Moore, and today we're doing a Science Live for Me, where we go through the journey and career of a scientist at the Bioeconomy Science Institute. Today we have Dr. Linley Jesson, who is a principal scientist in the data science group. Linley, welcome to the show.
Good morning, Tom. How are you?
I'm well, thank you. So before we all get started, could you please introduce what you do at the Bioeconomy Science Institute, where you kind of sit and where you fit in?
Okay, so as you said before, I'm a principal scientist in the data science group, and so primarily working on a lot of interesting data science questions, mainly at the moment around genomic selection, which is a technique that you use to, when you take the genomes of a plant and you sequence the genomes of a plant, you can predict what the phenotype will be. And so this technique can be really useful to predict what useful commercial traits that we have for a plant before the plant can actually exhibit those traits. So we could take 6-week-old seedlings, we could take the DNA, sequence the DNA, and as a result predict what the fruit characteristics of that plant might be in 5 to 7 years' time. So I'm doing a lot of work with that currently, mainly in kiwi fruit, but there's also applications in snapper, apples, rubus, berries, and lots of other fun crops as well.
That's really cool. So it's a very wide-ranging set of crops and fruits that you can kind of apply this skill set to.
Yes. So genomic selection is applied worldwide across all sorts of agricultural and horticulturally important things. So if we think about livestock and breeding cows, for example, they've been using genomic selection for the last 20 years to really accelerate the breeding of the New Zealand herd. And so these techniques are quite general, but are a really good way to accelerate crop breeding or animal breeding, depending on what you're doing.
Yeah, it's a fascinating area. And so I guess this podcast is about like your journey in science. So did you think that you'd be in this space when you first started out? Is there like a notable point where you thought I would sort of delve into science and begin this career? And was the kind of breeding pipeline and the genetics and selection and breeding program, was that in your mind when you first started out, or is that a completely far off concept?
When I was 7, I wanted to be an ornithologist. and I thought that studying birds would be the most amazing thing. And so I had a little diary, and I would write down all the things that I saw birds doing. And then I got bored of that pretty quick. In terms of moving into plant biology, that was more at the university level where I started off in general science, so just did the standard courses in first and second year. And then second year, we actually did a course, this was at Auckland University, on crop plants. And I just really enjoyed it. It was such a fun course, and we did a lot of hands-on work, sort of looking at the genetics and looking at some of the really unusual ways in which genetics of crop plants can work, and sort of looking at chromosomes and actually looking at chromosomes like down the microscope. And it was a really great course. That was Brian Murray at the University of Auckland who did that course, and that was just so much fun.
Yeah, I was going to ask if there was like a person that sort of you looked up to and was like quite pivotal and sort of directing you where you'd like to go. So this was that person.
Well, so that got me, actually from my undergraduate, which was in botany, I actually went and did a master's in environmental science, because clearly environmental science is a very important field. And so I did my master's in weed ecology. So that was at the University of Canterbury. And so I wasn't really flipping back into the crop breeding stage at that point. But in terms of pivotal people who've been really pivotal in my career, when I went down to the University of Canterbury, I got a part-time job reading for a professor of the botany department at the time, who had, so he was blind and tetraplegic. And so if you think about it, it was a terrible incident where it was thought that David was poisoned with acrylamide, and there was a trial at the time, and the person who was on trial got off, and so never convicted or anything like that. But the hypothesis was that he'd gone through some either poisoning event or some event to make him blind and tetraplegic. And so if you're blind and tetraplegic, if you're thinking about that, effectively in a box, right? You can't touch, you can't feel, you can't, you can speak and you can hear, but your interactions with the world are really, really difficult. So I got a job reading science articles to him. David Lloyd was a really respected professor of plant reproductive biology and worldwide renowned professor. And as a result of that, a lot of esteemed scientists came through to visit David because this terrible incident had happened to him. And so I got to meet some absolutely amazing scientists. And it was David who said to me, and this is a little bit of snobbery that I don't think we have anymore, but at the time he said, if you want to do a PhD and if you want to get a job in New Zealand, go overseas for your PhD. Don't stay in New Zealand. Because I think at the time we still had that sort of intellectual snobbery that PhDs from overseas were somehow more highly regarded than New Zealand PhDs. That's not the case anymore, I don't think. I would hope not anyway.
How long did you do the reading for?
For probably about 3 years. So I did my master's at Canterbury and then worked a little bit afterwards. But yeah, so I was reading for David for about 3 years, sort of 4 or 5 times a week.
And did the readings kind of give you a diversity of things that you could be pursuing in your future education, or was it like very narrow and you're like, actually I want to follow this kind of this field? Or
so David's field was plant reproductive ecology or biology, and so I'd go to the library back in the old days when you actually went to the library and go through the journals and pick out articles that we thought would be interesting to David. So generally in the field of plant reproductive biology. And David was great, and he would give me homework and say, here's a bizarre phenomenon in terms of reproductive of plants. Go away and give me some ideas as to the evolutionary mechanisms by which this might have evolved. And so a lot of it was also, it wasn't just reading, and actually most of it wasn't reading. A lot of it was talking and chatting and discussing, and yeah, it was so much fun and so enjoyable.
So in a bit of foreshadowing to your future career about in data, how did you actually explain the figures and the plots? Really
hard. And the other one was math, because David was a mathematician at heart, and so he came up with a lot of theoretical, really important theory about evolution of different plant sexual systems and different plant traits, but really with a basis, a mathematical basis behind it all. And so trying to explain math I just found really hard, really, really difficult, because you can't see, so you can't see the equations. And so if it's a complicated equation, you can't really sort of get the nuances going. So, yeah. So explaining graphs and explaining math I found very, very challenging, yes.
I bet, yeah.
Yeah. So as a result of that, David had said to me, go do a PhD overseas. And one of the people who came to visit David was Spencer Barrett, who is a plant reproductive ecologist, biologist at the University of Toronto. And so there was a dinner with Spencer one night, and I sort of chatted to Spencer on the way out the door and said, well, what's the possibility of me doing a PhD in your lab? And he said, go get some money. So I applied to the University of Toronto for a scholarship and got the scholarship and wandered off to Toronto. And so I spent 5 years at the University of Toronto doing my PhD in plant reproductive biology, looking at a bizarre floral syndrome of mirror image flowers where the female sex organ and the male sex organ of the flowers are deflected either to the left or to the right. So you might have the female sex organ deflected to the left and the male sex organ deflected to the right. And this might be on the same plant, you might have alternating left and right deflection of the sex organs. Or we've also showed that it can be a genetic polymorphism, a genetic effect, where in some plants are left-handed and other plants are right-handed, similar to humans. But it's the deflection of the sex organs that is under genetic control.
And did you have a hypothesis about what or why that was the case? What evolutionary processes was leading up to that? Is there insects or birds or...
So this polymorphism tends to be most pollinated by bees. There are other insects that do it, but often bees, and it's often accompanied by vibrational pollen collection. So things like tomatoes and blueberries have a system what's called buzz pollination, where the anthers need to be vibrationally milked by a bee to get the pollen out.
So do the bees literally go onto it and then start shaking it?
They start shaking it, and they use a specific pitch, and the specific pitch resonates with the flower, and that helps getting it out too. We've done lots of field work on this, and a really good way to get pollen out is to use an electric toothbrush and to vibrationally touch the electric toothbrush to the flower to get the pollen out. So the left and right-handed flowers, the idea is that if a bee comes in and it's getting pollen out from the anthers, and it might be vibrationally collected or not, the female sex organ will touch one side of the bee, and the male sex organ will touch the other side of the bee. And so then if it goes to another flower, if it has the same orientation, there won't be any self-pollination between flowers on the same plant. And so that's the idea for why there's this genetic one where flowers or plants are genetically left-handed or right-handed. The conundrum was the ones where they can alternate or they can be randomly distributed on a plant, so they can be left and right. And the idea that we had was that even still that reduces the amount of self-pollination that's occurring. It's not zero, but it's less than what it would be if the male sex organ and the female sex organ were close, so that every time a pollinator visited a flower, self-pollination was possible. This is a syndrome that Darwin was very interested in, and 6 days before his death he actually wrote away to ask for seed of solanum, a particular species of solanum, because he wanted to study it some more. So Darwin could have got there, but unfortunately he didn't live long enough.
Do we have any of these species in New Zealand that have that kind of reproduction?
Not native ones. They are ones that occur. So St. Paulia, so African violets, have a deflection of the female sex organ, but they don't have a deflection of the male sex organ as well. They just have the female. There's a species of Wakandorfia, which is in the Botanic Gardens in Christchurch, because I've seen it, and also in the Botanic Gardens in Auckland, and I've seen it growing wild on the side of the roads on the west coast. And in New Zealand, we only have lefts. So this is a genetic polymorphism, and so the flowers are only left-handed for any ones that I've seen in New Zealand. So the right-handed morph was never introduced.
Is there like a landscape variation that you can see? Is there certain areas that have left or right, or the certain combination of left and right, and does that change over like these landscape gradients at all?
It does depend on the species. So a lot of my fieldwork for my PhD was in South Africa, where this Wakandorfia occurs. And in Wakandorfia, there's a whole spectrum from plants where there's one-to-one ratios of these left and right-handed plants in a population, and that occurs sort of across a landscape gradient. But there are other ones where maybe the pollination hasn't been as good or as accurate, and so over time they've evolved selfing. And so over time, one of them in particular become a delayed selfer. So over the day, after the flower opens, initially it will start with quite a large gap between the female and male sex organs, and over the day that will lessen and lessen and lessen until it touches by the end of the day. And then if you look at ratios of left and right-handed plants in that species, it can be very, very skewed, because if you're a left-handed plant and you're primarily being pollinated by right-handed plants, if you're very rare in the population, there'll be quite a big advantage to being a left-handed plant, and so the theory goes that there should be more lefts as a result of that, and that would balance the sex ratio. If there's a right-handed plant and it's very common, and then it doesn't pollinate with left-handed plants very well, if it selfs, there is still an advantage to that selfing, given all else. So outcrossing is beneficial, but if you can't outcross, then there is an advantage to selfing at the end of the day. So in some species, you do get these quite skewed morph ratios because there isn't a strong selection for the equal one-to-one ratio, and in other species where they're much more outcrossing, then you get that strong one-to-one. That sort of fieldwork is really fun because you drive around the countryside looking for populations of plants, and then you walk around counting lefts and rights.
And do you also go out and collect the bees? Like, is there different sort of bee species that pollinate these plants?
Yeah.
And do they compete? Are they in evolutionally linked to these plants? Are they trying to attract certain types of bee species? Is it just the common bee species? Like, Yeah.
So the species, the Wakandorfera I'm talking about, the most common pollinator was the carpenter bee. So one of these really large carpenter bees. But there are other sorts. Long-tongued flies will also pollinate, and there are other ones as well. But yes, they do need to be a certain size, because if they're not a certain size, then the sex organs won't touch and they're in the right place. So yes, there is quite a strong relationship between the plants and what's happening with the plant reproductive biology and the bees. There are other plant polymorphisms, for example, where a plant might have a nectar spur or something where the adaptation is for things with longer tongues. And there are lots of floral reproductive polymorphisms. So one is heterostyle or tristyle, where you've got different lengths of the sex organs, not lefts and rights, but talls and shorts or longs and shorts. And those are adapted for, or have been quite specialised towards pollinators that have long tongues, so butterflies and hawk moths and things like that.
That's so fascinating.
Yeah, it's an amazing world.
Yeah. Just want to touch on the sort of people part of it. You did your fieldwork in South Africa, so did you interact with the communities there, and you sort of work alongside to sort of do the fieldwork? And
South Africa was an interesting time when I went there. So I was there in 97 and 98, so 3 months in 97 and 3 months in 98. So collaborating with people at the University of Cape Town. And there were also people at University of Stellenbosch as well who we collaborated with. But a lot of the work was just sort of driving around South Africa looking for plant populations and things. And it was a very interesting time, because that was just Mandela was president, apartheid had fallen over, and so a lot of these places where these plants were growing, there'd be quite large homeless communities there. And over the 2 years that I was there, it was actually quite interesting to see the changes that occurred in South Africa. So in the first year, for example, a lot of the homeless communities, there'd be an area where they would use as their toilet area, and you'd work out where that was and avoid it. And with the second year, I came back, quite a few of these places, there'd now be a toilet, there'd now be a tap, there'd now be sort of a bit more facilities. And so, it's a bit of slow growth there. But I did have a few situations. For example, I think one time I was wandering through, and there was a group of homeless women gathered around, and I was looking. I just said, "Look, because they'd say to me, I see you sitting in the flowers," and so I was just looking to see what they were doing, and they had a razor blade, and they were taking out stitches out of a woman's chin, and so she'd obviously been badly beaten up and been stitched up, and they were trying to remove the stitches. And of course I had alcohol and a bit of a cleaner scalpel and things like that. So I was like, wait, let's do this properly. And so I ended up taking some stitches out of a woman's face. And then they started showing me all their other bruises and illnesses, and all that was a bit shocking. But it was a very interesting time in South Africa, because it was, it's still a society that's coming out of apartheid, and it takes a long, long time for that to happen. Mm-hmm. Mm. But other parts of the fieldwork that I I wandered around central US looking for solanums, and that was a very interesting experience for a Kiwi with a very strong New Zealand accent. Wandering around Missouri and Kansas, looking for solanums, and I also did fieldwork in the north of Australia. And so there, the plants we were looking for were aquatics. They were in billabongs, and as was pointed out to me by the Australian academics, the monocoria that I was looking for were very favoured by the wild pigs, and the wild pigs really liked the monocoria plant species that I was working on. And the crocodiles really liked the wild pigs. And so here I was wandering through billabongs, not knowing if there were crocodiles there, but assuming that they were. So that was a bit scary.
I was going to ask, based on the previous location of fieldwork, did you have to carry a rifle to protect yourself from all the animals that potentially were roaming around the area? And, that was fine?
No, I never did. Watching for pollinators, because, as you were saying before, it's all about the interaction with pollinators. And so, especially in South Africa, where lots of these things were swamp-based plants. And so I'd be sitting on a rock in the middle of a swamp, in the middle of somewhere in South Africa, and I'd be like, oh, look, there's a snake over there. oh, that's nice. Oh, okay, I know where that snake is now. And then because you'd be sitting quietly, you'd be like, oh, there's a snake over there too. Oh, now so I know there's a snake there, and I know there's a snake there. And then you're like, oh, okay. and then you start realising how many snakes there are in South Africa. So, and my snake biology is pretty poor, so I just assumed they're all bad.
Oh, yeah.
But I definitely have seen a couple of spitting cobras there. So
that's really cool. At any point during your PhD, did you regret going overseas to do it, given the advice you were given?
No, I mean, the experience to go overseas and to explore new ideas, meet new people, put yourself out there in a new way, is really good. It's a really fun thing to do. I'm not saying that people shouldn't stay in New Zealand to do their PhDs, but it's always, I think, important to try and expand your horizons as much as you can and open yourself up to new ideas and new things. And so it's an important thing, I think, if you get offered an opportunity to take it, because you'll always regret what you didn't do. You very rarely regret what you did do, unless you do something against your morals, and don't do that. But I really enjoyed living in Canada when I was there, and to be fair, my partner's Canadian, and so we went back to New Zealand for a bit, but then we both got jobs in Canada, so we went back to Canada for 10 years. And we were in Canada in New Brunswick for 10 years before I went to Germany, and then going back to New Zealand. Yeah.
So when did you meet your partner?
So Dave was doing his PhD at the same time as me at the University of Toronto. So he was doing his PhD in plant physiology. And poor PhD students just spend all their time working and never get out. So the only people they get to meet are other PhD students. So, yes, we've been together now since, probably for 25 years, I would say.
So has Dave ever accompanied you on fieldwork to these exotic places?
Yeah, he did. We had a great collecting trip around Northern Australia where he was collecting things with interesting physiological biology, and I was looking for these monochorias. And so he would be the one standing at the side of the billabong as I was wandering through collecting flowers going, I don't know if there's a crocodile in there that's about to eat her.
So was Dave on crocodile watch, is that what you're saying?
Dave was on crocodile watch, yeah.
Yeah.
I'm not sure what you could do.
So I want to touch on that switcheroo between Canada, New Zealand, Canada. What drew you back to New Zealand, and then what drew you back out to Canada again? Was that the jobs, was it? Or?
Yeah, so when I finished my PhD, I got a job as a lecturer in biology at Victoria University in Wellington. And so I was on faculty there for 4 years. And at the time, Dave was doing his postdoc. So a postdoc is sort of a research fellow that you that you do as a fixed-term contract research. And he was doing that at Massey. But of course, if you're on a fixed-term contract, you need a permanent job. And so Dave was offered a job in New Brunswick and negotiated a spousal position for me. And as a result of the negotiation of the spousal position, I was told that I had to apply for a woman's NSERC award. So NSERC is the Canadian science granting body. They had certain awards for early career for female scientists to sort of push them forward. So I applied for this woman's award and was given that. And so that was quite a lot of money that came with that award. And so we went back to Canada for that. And so we were on faculty in Canada for about 10 years. And then, so Dave decided he wanted to leave academia. Academia wasn't for him. And so at the time I said, okay, well, if you want to leave academia and you don't know sure what you want to do, I'll apply for an award to go to Germany and we can go to Europe. And you can go find yourself in Europe because that's what we all do, right? We go to Europe to find ourselves. So I applied for a fellowship to go to Germany, which I got. And so I was in Germany for 2 years. Dave then decided that he wanted to become a golf course greenkeeper instead, and so ended up working on a golf course in Canada and would come to Germany for the winters because you can't play golf in winter in New Brunswick because the snow is too deep. So no golf there. But yes, so then I was in Germany for 2 years looking at genomics, studying genomics of mosses.
Okay, so we've gone from 7-year-old birds, no good, a bit of weeds,
plants, flowers, mosses. Yeah, okay. Mosses are really fun. Mosses are awesome.
Tell me about mosses.
Okay, yeah. Phil Garnick-Jones is. I want to have a shout out to Phil Garnick-Jones. So Phil was a very important mentor of mine when I was at Victoria University of Wellington. And Phil had a very good career at Manaki Whenua as a taxonomist. He was one of the authors on the Flora of New Zealand, Volume 4, which was weeds, but he became a professor of botany at Victoria University of Wellington. And so when I joined Vic, we would co-teach several courses together, and he was, and we were both very interested in plant reproductive biology. And Phil had started to get interested in mosses, and because mosses have really interesting sexual systems as well. So mosses can be male, genetically female, or they can be hermaphrodite.
Okay, and they can't be left and right mosses.
They're not left and right mosses, unfortunately. That would be kind of cool if they were. But the hermaphroditism seemed to be associated with a round of chromosome doubling. So Phil has sort of noticed that often you get what's called polyploidy, where you get whole genome duplication, and then this seemed to be associated with the hermaphroditism in mosses. So Phil and I put together a grant, a Marsden grant, which we were awarded, so we could get a postdoc, and we had a PhD student, and we started looking at this a bit more closely. And so as a result of this, when I went to New Brunswick, I kept working on mosses, on the evolution of mosses, and this correlation with genome doubling. And so that's what moved me into the genomic space. And so when I went to Germany, I really was looking at the genomics of a group of mosses where you had both male and female, so dioecious is what it's called, or hermaphroditism, and looking at the evolutionary transitions between what was going on there. And it did seem to be that the genome duplication had meant that it wasn't just a genome duplication, it was actually a hybridization event between two different species, and that caused the genome duplication. And that hybridization event, followed by the genome duplication, gave rise to both male and female chromosomes occurring. And that does occur in New Zealand. That species of moss does occur in New Zealand.
Okay.
Yeah.
But I presume that wasn't what brought you back to New Zealand after the stint in Germany. Was
Yeah.
that, or was Germany not the last point? Did you go somewhere else before coming back to New Zealand?
No, I went back to New Brunswick briefly. We had just rolled out a new biology curriculum, and I wanted to see that embedded in place. So I went back to New Brunswick for about a year. But at that point, Dave was a golf course greenskeeper. You can't be a golf course greenkeeper full year round in Canada. And my mother was getting older, and so I said, let's look at going back to New Zealand. And so that's when I applied for, as it was at the time, a job at Plant and Food Research, because I thought, okay, I might have a bit of an advantage here. So I applied for a job as a statistician. I had a background in plants, so I felt like I could talk to scientists really well about the plants and understand what was going on there. And as a result of all the work that I had done, most of my work was very, very stats and math-based. And so I felt that the stats background that I'd had as a result of all this work would be strong enough to allow me to work as a statistician. And so that's what I did.
So that skill set would allow you to translate between what the scientist's kind of worldview is, and then also about what the data can do, or the modelling can do, and then sort of bridging that gap between the two worlds and generating insights.
Exactly. Yeah. The nice thing about evolution genetics and genomics is a very statistical world. So basically what you're doing the whole time is you're working in statistics. It's just with a slightly different terminology, but it's very, very heavy statistical. So being able to translate that into what I'm doing now, especially around genomic selection, is a very easy transition.
So just quickly, you've moved around quite a lot within New Zealand, within the PFR or BSI. What do you consider to be your home site? I think this must have been like 5 different places you've been.
So when I arrived, I was at the Lincoln site, which is where we are currently based right now. I was there for about 2 months, and that was just to sort of become acquainted with the practices of the other statisticians at the time and to get used to them. I then moved to the Hawke's Bay site, and we were there for a few years, and was doing a lot of work on apples in the Hawke's Bay site, and not genomic selection of apples, but breeding of apples. So helping the breeders do that. I then moved up to Auckland, and that was as my mother was getting sicker, so we moved up to Auckland for that, and it was worked out of the Mount Albert site. And then Dave got a job in Bay of Plenty, So we moved down, and I was based at the Te Whaki site for a couple of years, and then it was actually remote working for the last year out of Te Anau, just which was very nice of the BSI to allow me to do that remotely. But now we've moved up and we're back at Lincoln, so let's call Lincoln site my home site now.
Okay, that sounds great. I'd like to note for the listeners that Linley arrived in Lincoln just before the major floods came through. So do you want to tell that story about? We bought a house in Little River on the Ukuti Valley, and we arrived on the Friday, and the floods from the Little River happened on the Monday, Tuesday. And so, yes, we met our neighbours very, very quickly, and our neighbours are very lovely people, but yes, it was quite a large flooding event that happened. So, yes, our neighbours did start calling us the rain people for a while. I guess reflecting back from this point, was at any point there was some sort of certainty about the direction it was going, or did it always feel like a sort of a choice of many paths, and you didn't really have an idea about what the next path would lead to and where the next decision point would be and that kind of thing? Like, was it a structured, purposeful career that you've led, or was it kind of ad hoc and kind of you just based decisions on where you're going to go based on what was in front of you at the time?
Yeah, definitely no planning. But what I like to say to other people is identify your maunga, and every time you make a decision, walk towards your maunga a little bit more. So work out what your mountain looks like, and where would make you happy. And every time you need to make a decision, move slightly closer. You don't have to climb the mountain straight away, but just move closer to it, and eventually you'll get to your mountain. And I think that's the important thing. So make the choices of the things that make you happy and keep you interested.
Yeah, that relates quite strongly to one of the questions I love to ask, which is if you were to give advice to a younger version of yourself, what would it be? And it seems
do more math.
Do more math.
Do more math. Stay in school and do math.
Would young Linley listen to that though?
No. It's one of these things now, right, with the AI, the whole AI revolution, etc. And you're tempted to say, "Oh, AI can do the math," but no, you actually have to have a good understanding of what's going on. You can't just trust that someone else or something else can do that properly. I think a really fundamental understanding of mathematics is so important in everything we do. So stay in school, go to math class.
Sounds great. I would put a vote for that as well. I suppose sort of on wrapping up now, looking towards the future, where do you see your career going? Where do you see your position is now? What value do you want to provide? Where do you see yourself, in the next few years?
Yeah. So as I said before, I'm a principal scientist, so that's a very important position. Not an important position in terms of the day-to-day job, but there is a role of mentoring, I think, as a principal scientist, and there is a role of supporting and bringing in early career researchers and making sure that people's career pathways are developed and able to be opened up. And so, I think any principal scientist within the BSI would say the same thing, that the role of mentoring and the role of supporting and helping others is probably much more important than their day-to-day expertise on their science career. So I think a lot of what I've been trying to do. I've co-led a few projects with various people, with early career researchers. I'm currently co-leading a taonga data project where we're thinking about the sort of governance of data that is taonga. So that of interest to Māori, and they might want sovereignty over that data. What would that look like? And I'm currently co-leading that with an early career researcher who's just such an amazing woman. So that's been really, really positive and rewarding. And I don't really think I need to help her much because she's pretty solid. But I think as a principal scientist, I think there is a role always to make sure that people coming through the science pathways can develop to their full potential as much as they need to.
And through that development of ECRs and your mentoring, is the landscape quite different now to when you went through all those years ago?
Yes, it is. So the landscape now is harder than it was when I went through. Funding is harder, and there are less opportunities for scientist-based driven research. So at the time, it was all very about scientist-driven research, hypothesis-driven for an idea. So things like Marsden, were based on that. Nowadays, people are moving more to missions and to sort of trying to solve big picture questions, which is an effective way to get science done, but really hard for an early career scientist to really pursue their passion. And so, as a result, trying to get opportunities for early career researchers to lead projects, I think, is a bit more tough. But I remain hopeful that we continue to do so, because if we don't give good paths to our early career researchers, we are shooting ourselves in the foot as a society. So it's a really important one. I'm also very, very hopeful that right now funding is hard in science, and that's worldwide, but I'm very hopeful that these things are swings and roundabouts. And so while we might not be investing as much currently in science, the role that science plays in society is so important that I think these things, so swings and roundabouts, I'll come back. So that's my hope. I remain optimistic about science anyway.
Thank you very much. Linley, thank you so much for coming in today and talking about your career and journey through science. It's been absolutely fascinating to learn where it all started and what things were discarded and kept throughout the way in terms of the domain and the weeds and the birds and where you've been throughout your entire career. It's been truly interesting. And I'm really interested to see what happens in the future with your career and how we can develop the next generation of scientists and what the landscape looks like in the future. So thank you so much for being here today.
Thank you.
And thank you out there for listening. If you want to hear more of our podcasts, please check out our SciGest podcast page or subscribe to our channel in iTunes or your favourite podcast provider. And don't forget to post a review and rate this podcast. Why don't you give it 5 stars? Mā te wā, and until next time.